Thanks everyone for joining this optical educational series. Dr. Ulrike Boehm is the speaker for today's ninth educational lecture. Ulrike got her bachelor degree at physics at the Technical University in Munich and her master degree on Cryo microscopy at the Max Planck Institute of Biochemistry. Afterward, she joined Stanford Hill's lab to pursue her PhD, while she focused on combining two advanced light microscope techniques to achieve isotropic diffraction and limited resolution in living cells. After her PhD, she joined the NIH in Bethesda as a postdoc to develop imaging and biomolecular techniques to understand how gene expression works. Currently, she works as a research specialist at the Advanced Image Center at the Janelia Research Campus. Here, he builds and modifies advanced optical microscopes, including lattice light sheet microscope and palm SIMU and so on. She consults and assists visitors all around the world on AIC instruments, data management, and analysis workflows. Moreover, I want to emphasize, Ulrike is very passionate about science community service. She has been highly engaged in community building, outreach, organization of workshops, teaching for more than 15 years. And today he will talk about single molecule microscopy. Ulrike, please take over from here.
Yeah, so thank you very much, Jen, for the kind introduction. Just only a minor correction. I unfortunately don't have a bachelor's. I studied in Germany during a time when there was no bachelor-master system. So I have only a diploma in physics, but I, yeah, just as a very brief comment. But independent of this, let's actually dive into the presentation. But before I start, I maybe would like to give you a very brief heads up about the fact that this presentation is actually a presentation which I merged out of two presentations. So it will be quite comprehensive. I'm pretty sure that you will learn a lot. But as I said, you will be overwhelmed with quite some knowledge. So please fasten your seatbelts for the next 60 minutes. But I also would like to just stress one part. It would be great if you could maybe save your questions for the end, because I'm a little bit worried that with too long questions in between, we might not managed to go through all the slides. So as I said, this talk right now is separated into two parts. The first part is more about the basic principles of single molecule localization microscopy, one of the super resolution microscopy techniques in the world. And the other part is more focused on the recent advancements of single molecule localization microscopy here at Janelia, which have been realized here at Janelia. But let's start with the first part about the basic principles, and also which also should give you some kind of practical hints about how you should kind of structure your single molecule localization microscopy experiment. So as we just again going a little bit back, as we are all familiar, there is a so-called diffraction limit in microscopy, and this so-called diffraction limit actually means that we are unfortunately not able with conventional microscopes to focus down with a normal lens down to a single spot. Instead, what actually happens is when we try to focus light, we will actually end up with something which looks like this. It's kind of like a blurred spot, and that's due to the fact that light, due to its wave properties, will diffract in the center of the focus, and therefore we will unfortunately not be able to reach an unlimited small spot. But instead, this spot will always have a specific extension, and the extension and the dimensions of this spot will depend on the wavelengths which you're using. So in the lateral space, it's approximately the wavelengths divided by 2, and along the optical axis, it's approximately double that size. So, and that in the end means that if we want to use visible light in order to see small details within cells, we will actually be hindered to see structures which are approximately 200 nanometers in size. But as we all know, biology unfortunately happens exactly at also length scales which are smaller than that, in particular when we would like to observe specific macromolecules interacting with each other. So, and the great thing right now about single molecule localization microscopy, or short SMLM, is that this technique actually allows us, enable us to overcome this diffraction limit. So as you can see here on the left-hand side, you have a quite blurred image of microtubules. And on the right-hand side, it looks like way crisper. You can actually almost see single fluorophores. And that's exactly what we would like to aim for in order to understand biology in a better way. So the basic principles right now of single molecule localization microscopy, or how this actually functions, is just pictured here. So what you see here is on the left side, you see a wide field image with the letters P, photoactivatable fluorescence microscopy, or PAFM. And in the lower right corner, you actually see a little bit of a sharper image. So that's the super resolved image. And in between, something happens. And what happens in between is kind of displayed here again. So what you can actually see is that instead of imaging everything at once, how we usually would do it in a normal wide field microscope, we actually only image in a single molecule localization microscope a subset of the fluorophores which are labeled in the sample. And that allows us, in principle, to very precisely determine the center from where the emission of a single fluorophore came from. And based on that information about the localization and its uncertainty, we can, in principle, when we add up all these signals in the end, we can reach or create a super resolved image. And that's in principle exactly what single molecule localization microscopy is based upon. And but there are actually different parts which one has to pay attention to in order to do this right. First of all, you have to make sure that you have the right hardware. So meaning the microscope which you're using needs to be set up in the right way. You have to make sure that the sample preparation is also done in the correct way, that you choose the right fluorophores, that the right fluorophores are also are optimized, or that the conditions for those fluorophores are optimized, meaning that you're choosing the right buffer. And in the end, you also have to make sure that you process the data in the right way, because as you can see here, it's not so that, unfortunately, like in a normal wide field microscope, that you only have one image, but yet that you actually take thousands of images, and these in the end need to be processed in the right way in order to, as I've mentioned, create a super resolved image. So in the following, I will just briefly go over the hardware. I will say a few words to the sample preparation, but then also to the processing before I talk a little bit more about how 3D single molecule localization microscopy is realized. And that's, as I said, only the first part. And the second part then of this entire lecture is entirely about recent advancements here at Janelia at the moment, as I've previously mentioned. But let's dive into the hardware part. In previous talks of this lecture series, you have already been made familiar with what actually a wide field microscope is and what also TIRF microscope is. And the basic principle on which single molecule localization microscopy is based upon is also such a type of microscope. So in a wide field microscope, as you maybe know, you can immediately take an image of your sample of interest, and everything is immediately captured on a camera sensor. So it's actually a quite fast way to take an image in comparison to the scanning approach. But unfortunately, with a wide field camera, you quite often also have a little bit of background. In order to avoid the background, what people usually also use in order to enhance the quality of single molecule localization microscopy images is they use the way to illuminate the sample via total internal reflection in order to actually get rid of quite some background. So as I said, those are the basic realization principles, but furthermore, you also usually have to spend a little bit of money into getting the right objectives, getting high powerful lasers. Also good cameras are necessary in order to capture as many emitted photons as possible. And then you might also need a bit more special optics if you want to go into the third dimension as well. So as I said, it's not like super complicated in comparison to other microscopes, but I think the complicated part comes when I right now talk about the sample preparation, and also when I talk about the processing part. So for the sample preparation, again, let's go to the image which we kind of already previously saw, and that's the blinking of the fluorophores. So usually this is not necessarily something which you encounter when you look at normal fluorescence microscopy data. So when you usually shine light on a stained sample, you will usually not see such a behavior, at least not to such an extent. Usually when you shine light on something, immediately you get the entire signal back, so everything is bright. So that actually means you need kind of special dyes, or you actually have to bring the dyes which you have in a condition such that they actually show this type of behavior, random blinking. And this random blinking, as I've mentioned, is usually not something which you experience with conventional dyes. In the case of a conventional dye, you excite a dye from the ground state to the excited state, and after a few nanoseconds, it usually relaxes back to the ground state once it reaches the end of its lifetime. But again, in the case of the dyes, or which you need in the case of single molecule localization microscopy, it's so that you actually switch that dye between something which is called like a dark state and a bright state. I have to give a heads up here because the dark state does not necessarily have to be a dark state. It only needs to be separable from the bright state. So it can also be a different color, but it shouldn't be the same color as the dye which you would like to, from which you would like to collect the fluorescent signal from. So as I said, so that's in principle what you want. And that's also why it took this technology quite some time until it could be realized. Because the idea for single molecule localization microscopy was already there a couple of years ago, even way before Eric Betzig and Harald Hess published their famous Palm paper in Science in 2006. But it was hard to realize because exactly this type of switching between dark and bright states was not really possible. So, but what are the exact properties of such a dye? So usually such a dye needs to be in the so-called on state for quite a bit. So if you randomly switch a few dyes on, you want that they are in an on state, and in that on state, they should actually emit as many fluorophores as possible. Then if they are back in the off state, they should also stay off, and because you really would like to avoid any kind of false signals. So if they are on, they should be quite long in the on state until they, let's say, for example, be bleached. And if they are off, they should really, as I said, not give any kind of signal in order to avoid like wrong counting of dyes and so on and so forth. Furthermore, it's also so that these dyes should have a high absorption coefficient and quantum yield, and they should be also resistant to photo bleaching, in particular because during such an imaging experiment, as I said, you do not image just only one image, but you actually take a couple of thousand images. You actually really would like to make sure that you collect as many signals from as many fluorophores as possible, and therefore these dyes need to be resistant to photo bleaching. But one thing also has to be said, that these properties, since they depend also on the laser power and on the buffer in which the fluorophore is embedded in, it's usually so that it takes quite some fine-tuning and optimization in order to get the conditions right to do a single molecule localization microscopy experiment. Right now, I would like to show you a few of the photoswitchable fluorescent proteins which are currently used, and also the switching mechanisms which underlays these type of fluorophores. So the first thing, I guess you might all be familiar with the photoactivatable GFP. That was actually the very first fluorescent protein which was also used for single molecule localization microscopy experiment. Here, what happens is the underlying principle is a decarbonation. Then another thing is, for example, lots of people in the single molecule localization microscopy field also use the so-called MEOS. As I've mentioned here, the transition doesn't happen from a dark to a bright state, but from one specific color to another specific color once you shine light of a specific color on these type of fluorophores. Then there are also other mechanisms, for example, cis-trans isomerization in the case of DROMPA, or even hydration and dehydration in the case of the so-called protein dry clunk. But of course, next to, beside the fluorescent proteins, we also have the so-called organic fluorophores or organic dyes, which are usually way brighter and also way more photostable. But again, as you can imagine in the case of fluorescent proteins, of course, you can embed them quite quickly into the genome of a structure, so they might be better for live cell imaging, whereas those, as I've mentioned, they are just way more photostable. Here's just like, again, an example shown with Alexa 488 or Psi3 and Psi5, which are very popular within the single molecule localization microscopy community. The only disadvantage with which the use of these dyes is that they need to be embedded in special buffers. So usually they need to be embedded in TIOL containing buffers, which again is kind of completely not suitable for live cell imaging. But they are bright, and they are usually currently at the moment at least the workhorse for single molecule localization microscopy experiments. Beside of these dyes, there are also the so-called spontaneously blinking dyes. So they recently were used quite strongly, for example, the so-called SIR dyes here. So they just blink spontaneously without any kind of weird buffer or without any kind of excitation from the outside. But beside of that, there you can also find so-called photochromic rhodamines. So these dyes have also the special property that they don't need any weird buffer, like for example in the case of the D-storm buffers I was showing before. Yeah, so current approaches for the realization of single molecule localization microscopy are displayed here. So they all have like quite fancy acronyms, but in principle they do the same. So I wouldn't say the same. I don't want that anyone of the community right now jumps at me or so, but it's so that the underlying principle is the same. So you try to make sure that there's only a subset active in order to be able to localize quite precisely from where the signal came from. But since the mechanism of the switching is a bit different, they sometimes also have to be treated in a bit different way when you do the analysis as well. And also the sample preparation looks a little bit different. But again, lots of different acronyms for kind of, I would say, the same principle, the same overall principle. Then let's dive a little bit deeper into the buffer conditions. So I mentioned to you that at least for the so-called direct storm approach, you use conventional dyes which you embed into buffers in order to make them blink, because under usual conditions, these organic dyes would not necessarily blink. So, but the problem is if you, for example, want to do right now multicolor experiments, it's sometimes so that you might have different dyes which emit in the optimum, or which have their optimal emission of their fluorophores or of their photons at different type of buffers. So what you can right now see here is like two diagrams, and these bars, you only have to remember the color, not the underlying buffer which is behind it, but you can actually see that all these different dyes were imaged with 5 different buffers. And overall, you can see the output or the total number of fluorophores which could be detected or which we were able to detect during these experiments. And you can quite clearly see that where one dye's optimum is, for example, the Psi3b has an optimum with the red buffer. Otherwise, the dye is actually quite low. But for example, the ATO647N has actually its optimum more with the green dye. So if you use the ATO647N with the green one, and you intend to do a 2-color experiment, you should actually not use the green one, because with that green signal, at least in the case of ATO I have to correct myself. I think Psi3b is still kind of quite good with that buffer as well. But the only point which I wanted to make with this graph is really that there are different buffers, they are optimal for different conditions. And sometimes if you want to do a 2 or 3 color experiment, you might actually more aim for a buffer which is not so optimal for a specific dye in order to optimize the conditions for the entire set of fluorophores. And this is, again, something which need to be tested. So if you get a new dye and if you are super excited to use this type of new dye, you always have to keep in mind that there are other relationships which need to be taken into account in order to move forward. But how do you test this? So dye, for example, can be tested in an experiment where, for example, here it's kind of quite nicely displayed. So here in this paper on the side, they actually test 3 different dyes. And what they do is they image the same structure or a similar structure with these dyes. And they just take a few thousands of images and they just check how many, how strong the signal is even after 50,000 frames, for example. And here in this specific study, what they also did is they even compared 2 different buffers. And this is necessary. This is actually really something, if you are not using an already optimized protocol, this is really something which I highly advise that you should do. So meaning if you aim for a super resolution localization microscopy experiment, if you don't know where to start, this kind of optimization step in order to see whether things are still blinking is really something which you should do unless you have an expert on your side who can precisely tell you what good conditions are or not. And then what I also would like to show, what this paper actually highlights, is that these buffers also differ in how stable they are over time in their pH. And that also can have a very big impact on how good you can actually take an image or how many blinks in the end you might be able to capture. So what's shown here is that these different buffers over time, over a period of up to 3 hours, they actually change their pH. And what happens is if they change their pH, that actually also means you change the environment of the fluorophore. And when you change the environment of your fluorophore, it might also be that you change their photophysical properties. And when you change their photophysical properties, you might in the end end up with less photons. So, and this is kind of shown here. This is again like a figure from that paper. So here twice we have taken images of similar structures taken within different buffers at like 2 hours after the buffer has been created. And what you can see here is that the buffer A, which is the GLEX, is way better in the better able to actually reconstruct the structure than it's the aged Glyox buffer. So, and that's really something which is quite important. So if you plan to do such an experiment, keep in mind that you always actually should use fresh buffer. If you don't use fresh buffer, you should actually at least be aware what the behavior of the buffer over time is. Will the pH of that buffer stay the same or will it change? And that can make a very big impact. So for example, if you don't see any kind of blinks, it might not be because of something's wrong with the laser or the hardware needs to be set up in a different way, but it could actually be that your sample prep is just not sufficient enough in order to make sure that you can provide the right environment for the dye in order to blink properly. Okay, and with that, we already dive into almost the last segment of the first part, which is the processing. And with the processing, I also would like to give a very brief heads up, and that's before we dive exactly into how we analyze the data, and that's about just the pure amount of data which we are actually handling and which we need to handle. So meaning if you have a strong computer which can perform operations on a specific image, you have to keep in mind that in this case here, you're actually dealing with tens of thousands of images which need to be handled. And that requires actually also a good computer. On the most ideal case, you also have a cluster close by, which lets you do the operation as well in a faster way. But this is really something which people quite often underestimate. And in order to maybe get a handle of this, let's, for example, say you take 50,000 frames with a 16-bit camera. And so you have a size of 1024 times 1024. That in the end would lead to over 1 million pixels. And if you have 16-bit, you in principle multiply the number of pixels you have by 16-bit in order to get to the full bit number. And that then approximately is 1 megabyte per frame. But if you take 50,000 frames, you actually have something like 100 gigabytes already. And that's actually lots of data to handle. And in particular, if you think about that, the fact that you actually do experiments in the biological fields, so you definitely need replica. It's not so that one data set is enough. You actually have to take multiple data sets. You also need positive and negative controls and so on and so forth. And then in the end, you might actually realize that your external hard drive can't deal with that data anymore. So this is really something which one needs to keep in mind, because when one does single molecule localization microscopy, it's really so that one actually works in a very big data sphere, I would say. But okay, that's just a very brief comment before we actually dive into the processing part. So you are already familiar with this slide, but right now what we actually do is we will talk more in depth about the actual detection and localization of these single spots, which we created because we had these special dyes, or we had conventional dyes with the special buffer. And once we have the localizations, we also need to render the images in the end in order to also do the quantification to get some numbers out of the data sets. But let's start with the first step. So the detection. So the detection is usually nothing else than at least the extraction of maxima in an image. So it's kind of like the coarse identification of where actually your single emitters are located. So that actually means you make lots of small boxes around your single emitters. So yeah, and this step can also happen via various type of different algorithms. Then the next step would be, once you have identified the single emitters in a coarse way, what you do is in the next step is you try to localize them way more precise. So you actually would like to know where exactly the center of the single emitters is. Unfortunately, this cannot really be determined super precise. So you can get a number of X and Y where approximately the signal of the single emitter came from, but you always end up with a specific uncertainty, just because it's just an estimation of the underlying position of that single emitter. And the so-called error, or the uncertainty of the position of that single emitter is kind of displayed here. So what this formula actually means, that so delta X is the uncertainty, equals or the squared of the uncertainty of the position of the single emitter equals the square of the standard deviation divided by the number of detected photons. And if we remember, hopefully I wasn't rushing too fast over the very first slide, how actually the ABI equation looked like for the diffraction limit, then we can actually see one thing quite clearly, that whereas before we actually had a very strong defined limit, which we couldn't overcome, because I said we weren't really changing anything with the microscope, so the numerical aperture stayed the same. And furthermore, we also used a very specific wavelengths, and we wanted to stay in the area of visible light. So that actually means that that's more or less a constant. Whereas this equation here clearly actually indicates that with the more photons we would be able to collect, the higher the lower the uncertainty, and therefore the higher the estimate of the single position could be. And that's actually, this gives us actually a way to overcome the so-called diffraction limit, which I've mentioned before. There are a few other papers out there which formulate the so-called uncertainty in a finer to a finer extent, where one also takes the so-called background noise, but also the pixel size into account. But this is, if you kind of go away from this presentation with just purely this equation in mind, that's, I think, all which like that's, how can I say, that's more than enough. But anyway, so, but then we actually end up with something like this. So instead of having in like a normal microscopy, a beautiful image, the end of the image taken and the end of the processing is in principle a list of localizations. And yeah, but right now I would like to go with you briefly through this type of list in order to for you to understand what's actually indicated there. So as I've mentioned, during the detection step, we have nothing else than we identified causally where the single molecule or the single emitter is kind of located. And then the next step, what actually happens is usually you estimate the position by, for example, fitting a Gaussian to that block. And in this case, it's just a Gaussian right now right through the center. And based on that, you can actually get quite precisely the X and Y coordinates in a 2D case. And those two parameters are also listed here as the first two columns within that list of localizations. Then what else do you have? Another parameter is actually the width of that Gaussian, or the so-called sigma. Then you have a few other fitting parameters, which is the intensity. So that's in principle the amplitude, the height of that Gaussian. You furthermore have the offset due to background, and you also have a parameter which is called like the standard deviation of the background as well. Then you have another parameter which tells you how good actually the fit is. This will be later on of importance whether you have multiple emitters or just a single emitter, and that parameter will also come in very handy in order to get rid of improper localizations as well. And then furthermore, you have the so-called uncertainty. So, and that's nothing else than the calculated precision which you can get out of a single fluorophore. So it's actually quite, I think that all makes quite some sense also based on what I've previously mentioned. But again, what one really needs to keep in mind is that the underlying data set right now is in comparison to a normal data set, not an intensity raster anymore, but as I said, a table. But over the years, it's actually so that the processing and quantification and also the analysis and interpretation of these type of tables is something which is actually quite I would say, something which is pretty fresh. So meaning the analysis of intensity-based or 2D pixel and 3D voxel-based grid data kind of was already there since the first CCD camera. But right now it's really so that the data format and how we handle this data is really something which is also a field of research which is also kind of pretty new, I would say, or has only been there for the last 10 years. So therefore, there's still lots of work in progress in order to handle these tables in the best possible way. But let's just talk about the processing and the different analysis steps. So right now, what we actually did is, yes.
Can I just quickly ask a question? It's about the sigma. In theory, should the sigma not be constant because it's determined by the aperture and the diffraction? So it should, as in, it should be a constant sigma for each blob. And if not,
in theory, exactly. So if everything's perfect, it would be. But the thing is also that the entire system, you have also aberrations within the system as well. And furthermore, as I've mentioned, like when you, for example, are in the position that you don't have a single emitter, but for example, multiple emitters, then you suddenly, but this is something I will get back to this in a couple of slides. I think like even in two, and then it maybe becomes a little bit clearer, because it really depends on how well you've set up the system. And but you're right. If it's a single emitter, you calculate it, you simulate it, and you know exactly how actually the response of your system would look like, and it should have a certain size. But sometimes it doesn't. So you actually have to how can I say it? It allows you actually to get rid of localizations which you don't want to have. But as I said, I say this in two more slides. Just wait a bit. Okay.
Awesome.
So
thank you.
Okay, perfect. So as I said, so we start out with 10,000 like 2D images, and then we transfer these kind of images into lists of localizations by the detection and localization step. What then happens in the next step is, and that's also what Juan mentioned, which I will also talk about in a couple of slides, is that you can actually kick out localizations which are not good, which are not that representative maybe your underlying understanding or model of a single emitter. So that's a step which is called sieving or filtering. Another step is then also you can do so-called drift correction, and you can also do something which is called grouping. For example, if a fluorophore is on over multiple frames, you can just like summarize these fluorophores as well. In order to also, you just do some corrections in principle with the image, but you can only do this when you actually have a very good handle on how the photophysical properties of your fluorophores are. So when you know these properties, you can, as I said, go in with the right correction parameters. And then in the end, you can either do the rendering, or you can immediately go from that leftover list of localizations. And that's also something which I want you to pay attention to. So you started up with 200,000, then you lower the number of localizations to 100,000, and then suddenly you only end up with 8,000. So with every step, you in principle get rid of things by just disregarding them, filtering out, or by just summarizing specific signals also together. And then in the end, you can also, from that more or less filtered signal of localizations, also go right into the quantification. And but again, let's just talk a little bit about the sieving and filtering of so-called high intensity or multiple emitters. So, and that's something which one was actually, I think, also a little bit referring to. So when we take just a normal cross-section through one of these single emitter signals, we would usually expect, also based on simulations, a very specific width of that signal. So we kind of know what to expect, because we know physics and we can simulate all these things. But suddenly, for example, if you take a cross-section through this bigger blob here, you suddenly recognize that instead of having a very narrow structure, you have a wider structure than expected, and which does not necessarily fit within your theory. But you actually know what the width should be. You know the constant which one was mentioning. So, and what you can in principle then do is you can either maybe find an algorithm which allows you to do something which is called multi-emitter fitting. So instead of fitting one Gaussian, you can just filter two Gaussians to your feature of interest. Or another thing is also you can look at the histogram of all these kind of signals. And based on the histogram here, for example, the histogram of the sigma, you get a distribution. So you have quite narrow ones and you have quite wide ones, but you know exactly based on theory that the wide ones are actually the ones which might maybe appear from multiple emitters. So, and what you can then in principle do is you can just set a very narrow range around the sigma, where you actually know that that can only be a sigma which comes from a single emitter, and you just get rid of the rest. So what this does is it allows you to keep emitters which actually have a very high likelihood that they came from a good from a single emitter. But what it also does is you also get rid of signal in general. You will later on see in the presentation in the second part, when I talk about the recent advancements, that there are ways to which still allow you in single molecule localization microscopy to actually also keep this more improper signals and still be able to extract very precisely exactly these two underlying positions. But as I said, I will talk later about this aspect in more detail. Then another thing, of course, one could also, in principle, filter the uncertainty. And I mentioned at the beginning that the uncertainty is nothing else than the blurriness of the single emitter. So if you right now reduce the blurriness, one could in principle assume that the more you reduce the blurriness, the higher the resolution will be. And that in principle, I mean, wouldn't this not necessarily mean that you can reach an insanely high resolution? Kind of, but one has to keep one aspect in mind, that a low uncertainty does not necessarily mean a high resolution. And why is that? Let's just look at this data here. So these are just like lots of crosses of localizations with their respective uncertainties attached as numbers. So, and if we, for example, say that we don't want to have localizations which are above 10 nanometer, because we want to claim that we have a 10 nanometer resolution in our image, what actually happens if we filter out uncertainties which are higher than 10 nanometers, we entirely lose the integrity of that structure. So meaning we can claim that we have the resolution, but it does not really make any sense, because you don't see the structure. So therefore, how can I say, one needs to be a little bit modest in what one does. I mean, you can only filter so much before you actually also lose the information in the image itself. So I hope that all makes sense right now. So you can go quite down, you can reject things which make an image blurry, but you also still would like to keep the structure in general. Okay, good. So that was about the sieving and filtering aspect. Let's just briefly dive into the drift correction. So with the drift correction, I actually wanted to highlight that. Okay, so what you can see here is that over time, while you are imaging, since you're taking 10,000 of images, it can be that the signal from one and the same spot, like here, for example, just a fiducial over time, will actually drift. And this drift will create a blurriness in your overall image. So you will see that there's a slight shift in the overall image. What you can actually do is, with so-called fiducials, but also with other strategies, this can actually be traced. And once this is traced, you can also trace it back. So there are ways to actually correct for these type of drifts. And this is also something which you need to do in particular, because you take so many images over time. As I said, there are different ways to do that, but as I said, I don't want to go into detail right now. So then there's another aspect, which is a so-called blinking correction, or also grouping correction. And that's a phenomenon which you observe that, for example, while you're taking images, it can be that over different frames, you see signal which kind of is still on over different frames. And with a so-called grouping time, which is a time which you would allow a fluorophore to be on, it's a very fluorophore specific time, you would be in the position to actually group fluorophores or molecules together. And therefore, you actually then, like, first of all, you don't have false signals, but at the same time, it's also so that you have an overall higher photon count for that specific fluorophore, and therefore also a higher precision. So that's a correction which you want to do. But this is also a correction which will lower the number of fluorophores or of detected signals in general. But again, it's a good correction. And then there's also something which is called like grouping in space. So this can also be done with a so-called grouping radius. So you can, this grouping radius then also allows you to, in principle, shift everything together, and you also have an overall higher certainty when you do also the grouping in space. Yeah, so overall, one can say that there are lots of software packages out there in order to do exactly these steps which I've already described, either the detection or the localization, or also other drift correction steps. Fortunately, over time, because I guess it's always so when there's a large set of different programs available, it's sometimes hard to make a choice. But fortunately, in 2015, but also 2019, the community kind of came together, and with a so-called single molecule localization microscopy challenge based on benchmark data sets, they were quite nicely in the position to compare the performances of these individual steps. So, and if you want, you can actually read these papers in more detail and look for your algorithm of choice. But let's just very briefly again talk about the timeline of single molecule localization microscopy in order to get a better understanding why there still needs to be lots of, why we still have to do quite a lot in the quantification of single molecule localization microscopy data. So first of all, the first single molecule images were taken in 1995. Then the first emitter were localized, or at least the theory for the localization was established by Thomson in 2002, and then later on as I said, these hallmark papers in 2006 about single localization microscopy were also just more or less quite recent. But what's currently actually going on in the community is an incredible rise of different ways to actually handle these different localizations and how to manage these different entries in the table in the localization tables, as I've mentioned before. So, and there are lots of ways to do this actually. There are strategies to kind of pool these things together, because you always have to keep in mind that you have in principle an infinite small spot kind of in space. And it's right now, when you have different spots which are kind of very close to each other, it's also very hard to understand the concept, for example, even of co-localization with just pixel clouds. So, and therefore, different types of algorithms and understandings and operations, how to actually deal with these type of localizations needed to be and still need to be invented. But this is just like a very brief overview of a quite nice protocol in nature protocols. Yeah, but beside of that, there are also lots of free software available, in particular also in Fiji, to, for example, also do cluster analysis and to handle these localizations also in more detail. But as I said, these all kind of came up quite recently, I would say. But and as I've mentioned, the community is still kind of actively working on handling these type of data. But with that being said, I will just right now dive very quickly, and I know that we are kind of running out of time, but over the 3D aspect of single molecule localization microscopy, before I also very quickly go over the recent advancements here at Janelia. So for the third dimension, one has to, again, maybe have a look again at how actually the point spread function along the optical axis looks like. And unfortunately, as you will realize from this scheme, that when you're 200 nanometers above or below, it kind of looks like the same. So meaning from a very, from this kind of point of view, it might be actually quite hard to determine whether a signal from where along the optical axis actually a signal came from. So in order to still be able to determine from where a signal came from, what happened over the last couple of years was that people thought about ways to actually break the symmetry. And once you break the symmetry, you will actually be able, as you will see here in the next slide, you will actually be able to suddenly see whether a signal came from above or below the focal plane. So what you see here is that if you pay attention to the number, while we go from minus a few 100 nanometers to plus a few 100 nanometers, that the signal actually at the same number on top and below is actually slightly different. So in the first case here for the astigmatism, we use a cylindrical lens. Here in this case, this is like a biplane approach where you actually detect different focal planes at the same time and get this asymmetry via that. And then we also have a strategy which is called a double helix. As you can see, while you go through that, you actually have a rotating point spread function where the position of the different axes is always slightly shifted, and therefore you can quite nicely tell whether you are actually below or above the focal plane. And then there's also another instrument which we have here at Janelia. It's the so-called interferometric palm. And I hope you can see this a little bit. Yeah, the scheme works. So while you're detecting the signal, you actually you collect the signal from two sides, you let the signal interfere, and you detect it at the same time on 3 different cameras. And due to the fact that you have slightly different optical paths, it's so that the signal which you detect in the end will, as you can see here in that scheme, will vary depending on where the flow for actually is in space. And you can nicely trace that back to with a very high precision to where along the optical axis the signal actually came from. So that's a quite powerful method actually because of the interference. Anyway, so let me just briefly summarize the advantages of single molecule localization microscopy. So it's a very powerful method, which allows you with a very, which gives you a very high resolution. As I've mentioned, also due to the name, it allows you to actually detect single emitters and single molecules. But you can also apply this in 3D and so on and so forth. But there are also a few disadvantages attached to the technique. And these disadvantages are, for example, that you need a special buffer, that you are usually not in the position to image very thick samples, that the acquisition is also very slow. And why is it slow? Because you really would like to be able to detect single emitters in order to be able to fit very precisely the signal. But a few of these aspects, and that's also something which I would like to highlight right now in the second part. And I promise the second part, I promise I won't run more than, I think the second part I can manage to do this in 15 minutes. So we run over by, I would say 10 minutes, but I try to do it a little bit faster. But anyway, but in the second part, which is nothing else than the recent advancements at Janelia, we will try to tackle a few of these aspects, a few of these disadvantages. So I will show you ways how one can actually do the acquisition in a faster way. And I will also show you something, how can I say, how we don't necessarily need kind of special buffers. And I will also show you other ways in order for you to understand that we are not necessarily restricted in thickness. And a few of these examples or these recent advancements came from different labs here at Janelia. So on the one hand, I will very briefly talk about the hardware aspects, which are developed here in the Harald Hess lab on campus. Then I talk about a few new dyes which have been developed for single molecule localization microscopy in the Localizations lab. And finally, I will very briefly touch upon a deep learning way in order to analyze the signal from single molecule localization microscopy measurements in order to actually do these measurements faster. But why this is the case, you will get to know in a very brief minute. Okay, let's talk about the hardware aspect. So, and this is work done by Gleb Stengel, Sean Xu, and also Harald Hess. So Harald Hess, as you maybe know from his previous work, is actually one of the co-inventors of the PALM technology, which I've highlighted. I think I mentioned this several times already, which was published in 2006, and it's actually quite powerful. And after his work on single molecule localization microscopy, specifically on PALM, his scientific interest still kind of continued to push the boundaries of the resolution. Specifically, right now, he invented in his lab also the IPALM technology, which I've mentioned as a way to extract 3D information, was also invented. And furthermore, he's right now actively doing his research also in correlative light and electron microscopy. But that also under cryo conditions. And that has actually a very big advantage, because when you do measurements under cryo conditions, what you will also see is that, what you specifically will see is that, first of all, you freeze the samples, therefore the sample's integrity is kind of still intact. And furthermore, it's also so that under cryo conditions, you are in the position, or you are able to collect way more electrons, because the photo bleaching is prevented, or at least not necessarily prevented, but the samples don't photo bleach that strongly. It's kind of nicely seen here when you compare these two graphs. So one is under room temperature, the blue one, and the other one is under liquid nitrogen conditions. So you can actually see that you can collect way more signal over a way longer period of time. And I think in total, you can collect 380 times more photons when things happen under cryo conditions than under room temperature conditions. So the Hess lab right now, that's like just one example. I hope my computer does not really crash anymore. But what you see here are two rows where at the top row, you actually see measurements under cryo-helium conditions. and the lower row are measurements, single molecule localization measurements under liquid nitrogen conditions. And what you can actually still see, so that's the main takeaway from that movie, is actually that the blinking still happens under cryo conditions in vitrified ice. And if we just go to the results, is that you can still, I'm sorry, that was too fast, that you are still able to very nicely get super resolved images out of this as well. And how exactly is this done? So this is the entire technology, the entire imaging happens in a so-called cryostat. So what in principle happens is you have your samples, you freeze the samples, you freeze your cells under a high pressure freezing, and then you transfer the entire sample into a cryostat where the sample is then placed in vacuum. And what in principle happens once the cryostat is within that entity, you can actually from the outside do, for example, structured illumination microscopy or even palm microscopy. And here's just like one example of a 3-color ZIM and 2-color single molecule localization microscopy image taken under cryo conditions. And again, I hope that my computer does not really crash. It's actually, as I said, quite remarkable because, as I've mentioned before, usually you would fix samples and with fixatives. These fixatives, they can do something to your structure, so they can compromise the integrity of your biological structure. You don't want that. But when you freeze samples quite quickly, and when samples are then kind of fixed in amorphous ice, the integrity is more or less intact, and they could show that, as I said, one can still do structured illumination microscopy as well as single molecule localization microscopy. So right now I only have to change to the next slide. Sorry about that. Yeah, sorry. That was right now a little bit too fast. Happens when you have large movies. But anyway, so the story hasn't stopped there. So they were not only able to do this under cryo conditions in order to prevent the that the substructure is not damaged, but furthermore, they actually even went one step further. So they were imaging the samples, but then they also transferred these samples into an electron microscope, specifically a focused ion beam scanning electron microscope, and they were in the end even able to correlate these type of microscopy images to with very high precision. And here's just like one image taken from a science paper in order to just highlight to you how well this actually works. So again, we are kind of flying here over cells while you actually can beautifully see the electron microscopy images and taken on the focused ion beam microscope. But when we then also go further into detail, one can actually see that they were also able to correlate this with the respective cryo palm data set as well in order to have the specificity. So that's usually also something quite nice, like, because we all know the advantages of electron microscopy. You actually have a good resolution. You can actually see things to a very nice extent, but unfortunately things do not really show up in different colors. So, and therefore we need something which kind of guides us to the regions of interest, and what in principle can guide us. And they were showing this quite beautifully in their work spearheaded by Gleb Stengel and also David Hoffman, that you can combine cryo super resolution microscopy quite nicely also with electron microscopy. But again, I will stop stop this movie right now because, as we are aware, I'm running out of time. So we just go right now to the next slide in order to, sorry about that. Okay, I just skip the summary for now, and we just dive very briefly into the second aspect, and the second kind of recent advancements I would say is that's like the development of new dyes for single molecule localization microscopy, spearheaded by former postdoc Fadi Shradi in the lab of Luke Levis. So and again, many of you might know Luke. I mean, Luke's lab is quite famous for the so-called Janelia farm, or like Janelia fluorophores. And for those of you who are interested in getting these fluorophores, using these fluorophores for your own experiments, please check out this webpage. Note this down, check this webpage out, because a few of you maybe might have already read the Open Chemistry eLife paper, which Luke recently published about Janelia's efforts also to make our chemistry available to the world. So if you need one of these amazing dyes, feel free to go on this webpage, register, and get whatever dye you need for your experiments. But again, just coming very briefly back to what I've mentioned before. So there are these D-storm dyes. These D-storm dyes are quite powerful organic dyes, which bleach less strongly than fluorescent proteins, but again, they have the weird property that they have to be used in these weird buffers. So on the other hand, it would actually, yeah. So on the other hand, there are other, like there are dyes which don't use necessarily weird buffers, which can be used, for example, just under normal physiological conditions, for example, just in PBS. But therefore they might also have very weird photophysical properties. So sometimes, at least in the early days of the invention of organic dyes, there was always a drawback. Either you need special dyes and have good, good signals, but or you have, how can I say, you have to compromise the photophysical properties of your dyes, but can then do this maybe under live conditions. But Luke's lab, they actually wanted to, again, push the development of so-called spontaneously blinking dyes, or also photochromic rhodamines, even further. So those are, as I've mentioned initially, dyes which can just emit fluorophores, but you can also in principle do live cell imaging. You don't need any kind of crazy toxic buffer or something like that. And they were actually able to come up with new dyes which can spontaneously blink, and or also dyes which have so-called photochromic properties. So they don't necessarily spontaneously blink, but you need an external wavelength in order to make sure that they emit a signal. And so just to the question, yes, they could, they were able to do this. And these dyes are right now based on so-called coumarines. And as I've mentioned, since they are so crazy bright, and they give off like so many photons, that they came up with this very creative name for this dye. And drum roll, please. So they call this dye glitter bomb, because it's just like an explosion of color, and like it just emits and emits and emits and emits. So so right now I think the glitter bomb 594 is actually the one which has been tested and quite well understood. I think the paper will also come out hopefully pretty soon. But as I said, this work has been spearheaded by also, yeah, by Fadi, also a good friend of mine, is a very great chemist, and based on this type of dye, then this was also tested multiple times, then by different people here, for example, combined GB, like stands for glitter bombs. So at a certain time, it should also be, I guess, on the web page, hopefully be available. Might not be available now, but hopefully soon. Here right now, glitter bomb is attached to phalloidine, and they were able to show that they can actually go quite down with the localization precision. And then also it was then later on also by Jesse Iron, a member of the Advanced Imaging Center, also shown in other structures that the precision even along Z can be reached quite nicely with these dyes. Okay, then just the very last aspect. And that's not the hardware part or the die part, but this is this time the software part, which also has been pushed here on campus quite heavily, in particular in the lab of Srini Turaga. So combined with different groups at the EMBL, he was actually able to solve an issue which also one mentioned at the beginning about the Sigma, and the problem which they tried to tackle was the problem that you usually need to take quite a few images, just like for the sake of time. So you usually need to take quite a few images in order to get such a beautiful image. But if you want to compress that, so meaning if you want to take less images and therefore be faster done, which also would be handy in order to, for example, use single molecule localization microscopy for any type of live cell imaging, you end up with the problem that you suddenly have overlapping fluorophores. And these overlapping fluorophores make it hard on your algorithm in order to extract the localizations properly. And that in particularly will be shown at places which are very dense. So the denser the places, the more the likelihood, the higher the likelihood, if you compromise in time, that you actually have two fluorophores which are maybe on at the same time, or multiple fluorophores. And these, under normal conditions, with normal algorithms, would just be disregarded. And therefore, it's quite typical when an algorithm cannot deal with a high number of single emitter signals, that you actually suddenly see that it becomes darker there. Usually it should be brighter, right? Because it's like you have more stuff there. But it's darker because the algorithms can't deal with that stuff anymore. So, and that's why they usually kick out these so-called insufficient localizations, and you end up with these very weird structures here. So, and Srini and his collaborators right now developed Decode, and Decode is nothing else than a deep learning based algorithm which helps you to actually overcome this problem. So because this algorithm right now specifically has been designed for high dense 3D localizations in single molecule localization microscopy. And it's not only good for high density, I mean, that's also what I said, the denser, the more dense a data set can be in order to be handled by that algorithm, the faster you are also able to do the imaging. And the faster you can do the imaging, potentially also you would have a chance to do live cell imaging even better than it was ever done before with single molecule localization microscopy. So this entire algorithm is based on unsupervised learning, and is only actually based on calibrated data. But I will very briefly say something in a couple of minutes how it actually operates. And another secret ingredient is also that it takes the so-called temporal context in mind. But you will also see this in just a minute, and I promise we will be done in time. But in order to understand this, I'm just very briefly diving back on how a normal processing actually operates. So usually, as I said, you would end up, or you would start up with your image, and then you go through whatever kind of detection localization algorithm step. So we talked about this before. So, and once you have decided based on your desire, which type of algorithm you want, you in principle end up then with like a set of coordinates and like background signal intensities and also uncertainties. And with a little bit of post-processing, you then end up also with a rendered image. So, but what also has been said here is that this entire part here for the detection and also the localization is something which is also very, I would say, stiff. So it does not really adjust whatever data set you have or whatever underlying conditions you have. So therefore, the difference right now with Decode is the following. So first of all, instead of just taking one image into account, you actually take the temporal context per image into account. And that helps you then actually also to figure out, how can I say, how to treat different localizations within one image. Because as I said, you have the temporal information. Maybe that also is kind of familiar when you just think about the grouping along the temporal space, which I mentioned at the beginning, that taking this into account can also increase your signal. And here, as I said, it's kind of like along the same line. And furthermore, it's also so that right now this so-called decode network, which is based on a convolutional neural network, is also something which, how can I say, which kind of changes according to which type of camera and fluorophore you have. So this is something which needs to be trained. And when it's trained, when it's kind of properly designed for your specific purpose, for your specific instrument, and for your specific fluorophore, it can actually do these type of predictions in order to get good single molecule localization microscopy data way better than it was done before. So as I said, the secret ingredient is this convolutional neural network, and furthermore also taking into account the temporal context. So, and how right now, and in order to run through this, you also first of all, of course, have to design this decode network in order to do the entire, to be able to do the entire flow. And how is right now this network designed? So this network is based up on simulated data, and this simulated data takes into account the fluorophore, the point spread function which your system has, the camera which you're using, and based on all these kind of preliminary data, what in principle happens is you simulate a bunch of data sets, and you feed these simulated data set into that algorithm, and then you get lots of predictions out. And as I said, this is the first part is something which you have to do. So you have to specify, you have to give a little bit of specifications about your system into the network. But then what happens is it kind of, how can I say it? It builds itself up according to your specific problem. So these predictions are then compared to the ground truth which you've put in, and based on then the loss function which you create, which is in principle nothing else than a function which needs to be minimized. So you want to minimize the error between what you've actually put in the system and what came out in the end. And based on finding the minimum of this loss function, you can actually, or based on ways to minimize this error, you can actually then shape the network in such a way that it's optimal for your specific problem. And again, that's the underlying signal, the underlying way of actually how to train the decode network. And then just very briefly, I would like to show you how the results then in the end look like. So they, Srini's team compared decode also with a bunch of other algorithms to extract localizations, for example, like C-spline. And what you can quite clearly see, whereas C-spline is not really able to, it is kind of able to extract the signal, but as I said again, once you have an area where you actually experience a higher density, you usually have a decrease in signal because the algorithm cannot usually handle overlapping signal. But fortunately, decode is actually quite nicely or way better in the position to extract the localizations, and therefore you actually don't see the dimming effect which you would see when you use, for example, the C-spline algorithm. And furthermore, for example, what you see here are again different data sets where actin has been labeled, and what you actually see here, every column is nothing else than a capturing of the underlying signal, and the longer or the shorter the amount of timers, the less signal you would expect. But in order to compensate the lack of signal, you actually increase the laser power. So, and therefore you would have a higher density. But as you can clearly see although you increase the density, it's still so that the images on the top row still look actually quite similar. That actually means that you can record super resolution localization microscopy images within shorter intervals and still have a very good result in comparison, for example, to again the C-spline algorithm, which is actually really struggling when you try to do the same recording within 93 seconds. Okay, so then at the very end, just like a very nice overview here of the performance in terms of detection accuracy and also localization error. So what's indicated here within that within those diagrams is actually how different algorithms perform. So every single spot here within these diagrams indicates a different algorithm, but the very thick ones here at the bottom is actually how decode performs, either in a 2D single molecule localization case, or when you try to analyze astigmatic data or the double helix data. And what you always want is you always would like to have a high detection accuracy and a low localization error. So you always would like to be in the very on the lower right hand side. And as you can see, based on the types of data sets, so the lower the signal and the higher the density, so that those are usually the highest challenges or the highest challenging images, you can actually see that decode quite nicely is always in the lower right, so therefore outperforms most of the algorithms out there in order to do proper localization. And then at the very end, just like one example, what you can actually get out of a recording, which only takes 3 seconds, you can actually almost see the nuclear pore quite nicely, even when you only image for 3 seconds, and which I think is actually quite remarkable. So again, usually you would expect that you would need to take a few thousands of images in order to get a proper reconstruction. But I would say this result is already a nice indication that we might actually be on a good way to do also even better live cell imaging with single molecule localization microscopy in the near future as well. Okay, with that, just a very brief summary, but since we are already out of time, like feel free to read the summary in your free time. But just a very brief point that you can actually also find tutorials and all the information about this type of algorithms also online. Everything's freely available. So same thing like with the dice. I think Janelia is quite nicely open science positioned, and I think we continue to also go that path. But with that, and with only, I just have to check the time, with only being 18 minutes over time, I thank you for your attention, and I'm open for questions.
Well, thank you very much, Ulrike, for fantastic. We've covered the whole breadth from top to bottom. I guess we have around sort of 10 minutes for questions and answer. I'll start off with a few questions. The first question is 3D localization. So one question from Chen would be the axial imaging range. What would that be? Type? What sort of axial imaging range would 3D localization cover? And secondly, for my question is, wouldn't shaping the 3D PSF also reduce the lateral resolution? Because you're exciting a lot of florals at the same time with a broader PSF.
Yeah. So yeah, but you're entirely right. So the thing is, when you at least with point spread function engineering, it's so when you deviate from the normal point spread function, which you usually have in the case of a confocal microscope, which sits in ellipsoids in space, you have actually the most compact accumulation, which more or less when you use a single objective, which you can get. So therefore, every change with that very compact design, let's say, for example, in a case of a double helix, or in particular in a case of an astigmatic point spread function, means that you spread out the signal over a wider range, and therefore you actually have per per slice, how can I say, less photons, which actually also means if you just think about the equation for the for the uncertainty, that actually means you actually have less fluorophores and therefore you have a higher uncertainty. And so therefore you're right. You might then, in principle, when you spread it out, you might actually need to accumulate the signal over a longer time, meaning you might actually need maybe a longer exposure, or you might also even need kind of better dyes with more fluorophores, or with a higher photon budget. But about the 3D or third dimension, so there are different ways how to do this. I think the group of W.E. Murner in Stanford, for example, they came up with the double helix, which is actually quite extended, even more extended than just like over a micron. I think it even goes almost to 2.5 microns. So that's quite impressive. But I also have to say that then you actually distribute the signal over a really very wide range. So meaning when you image one plane, you might be able to actually image like a very thick sandwich, but it might actually be better in terms of like photo efficiency. And that's also something which other groups do, to, how can I say, just have an asymmetric point spread function over a smaller range, but then stack these ranges. Sure. So meaning you image, for example, over 3 up to 400 nanometer, and then you drop the stage, and then you image again with and then you do this imaging again.
Right,
I think like
sequentially just stepping up the whole.
Yeah, and that's something which, for example, the group in Yale, led by Jörg Beverstorff, I think they've implemented this in their 4-pi single molecule switching microscope, that they actually, but that then again also requires another thing. You need to have a very good precision. So you need to make sure when you drop the stage that you actually always how can I say, that you can actually merge these different layers quite nicely together again.
Yep, sure. Any questions? Anyone else? Yep. You can type it in the chat, or unmute yourself to ask. Yep.
I have a question. So you're mentioning about how the buffer changes over time. But isn't it the case, is it just not the case that you don't have enough concentration of buffer, like say Hebes? Isn't it, can't you just increase the concentration of the buffer components to eliminate that change over time?
Yeah, I think but you're right. Like, I think there's media out there where how can I say, where you would have the ability to keep the pH quite stable, but at least also at the AIC, from the experience which I had when I was working at the interferometric palm, was really that I always got the best results when I prepared this always very fresh. So I don't know exactly what all these underlying conditions are, but we actually tried to have mixtures which were kind of very stable over time. But in general, because I'm always like a little bit cautious also about the fact that if you put quite some time into the sample prep, and if then also the data taking and the analysis takes quite some time, you don't necessarily want to compromise any of these things, because then you might actually have to do everything again. I guess, no, but I know, I hear you, I know what you mean. So you actually aim for the best thing. And I think that's also, for example, with this one buffer which was displayed, where you actually have also long-term stability, where that's the case. But still, I mean, I'm like, when I do experiments, I'm always on the more cautious field. I mean, things can be seen or said to be stable, but then I don't know, I always start with fresh ingredients just to be on the safe side. I don't know if that was a satisfying response, but I know what you mean. There are things which are stable out there, but I'm just like I don't know.
That's right. And my second question is, would the wavelength you use change your localization uncertainty? Because you said it's decoupled. Now we're decoupling the resolution from the point spread function, right? Because it's localizing to a single blink, photon blink. But if we, would this still be affected if we were using, say, a 488 nanometer laser versus a 647 nanometer laser? Because suddenly our or not.
Yeah, so the underlying, I think that's also a point which John kind of brought up. So the redder the wavelengths, the more extended the stuff. So
yeah.
so meaning based on the AB equation in general, when you have smaller wavelengths, you also have a higher resolution because you are kind of smooshing this into like a smaller volume. So yeah, and that also comes back to what you I think initially also mentioned in I know what actually Steve mentioned at the beginning about the 3-dimensional resolution. It's kind of like okay, we are not artificially changing this by for example a double helix or astigmatism, but even changing the wavelengths also makes things more compact. So you have in principle, you can expect in principle also more photons per area, and therefore you would in principle automatically also have a higher precision. But there's only so much room you can play with, because it's, I mean, the visible range is only like from 4 up to 780. So yeah, and it is also so like you might maybe, that's also the drawback of using lower wavelengths, right? You might maybe be able to use lower wavelengths and have a more compact point spread function, but then you're also more toxic. And lots of fluorophores, they cannot really handle these. They cannot really handle 2 blue dyes. Can only like, I just think back about when I did my PhD, we tried to do some kind of activation also with a 4-5 laser, and like after a few slides this was all gone, and then we kind of changed this to 2-photon excitation. So we went like double the wavelengths, we doubled the wavelengths, and therefore you actually have a way broader spot. But in the end, that was like way nicer for actually the fluorophore. So what is in theory maybe better for resolution might then compromise something else in your experiment. So one always has to be mindful that like one parameter change also might actually be very bad for other parameters during your imaging experiment, which needs to be kept in mind. Cool. But yeah, cool. I hope that.
Yeah, thanks.
Any more questions from the audience? If not, I'll end off with just the last question. The last few slides you showed was very impressive, where the correlative imaging between at low temperatures of single molecule imaging plus cryo. That seems very tough and seems very unique. And is there anything special about the sample prep, particularly to achieve that localization?
I mean, I think there are lots of things which, how can I say, when it's like, I used to work in a lab in Germany, which kind of started out also with correlative microscopy, which kind of tried to marry also fluorescence microscopy and an electron microscopy. And based on that experience, I can say, which is like more than 10 years ago, I have to say, by the way. But my experience from that time was always that, first of all, that the transfer is also quite tricky. So meaning that that's not like necessarily obvious, but unfortunately, I think there were attempts in the past where one could kind of marry a fluorescence microscope with an electron microscope, but these are not necessarily commercially available. So meaning once you've done the imaging in a fluorescent microscope, you somehow have to get that into the electron microscope without contaminating your sample. So I think in the case how Harald Hess and his team are doing this right now, it's so that they do the observation under cryo conditions, that the sample is in amorphous ice, but then they actually replace the ice with resin in order to actually do the electron microscopy later on. So they don't have the entire cryo flow, because the entire cryo flow, that's actually really something which I think would be something which I would really like to see in future, because then you actually keep the integrity of your sample all the way kind of intact. But I think that the special thing is really, just coming back to what you said about the special thing. So first of all, it's, as I said, it's not easy. That's what I wanted to express with my first response to the question. But it's also so that you need to have like long-term stability for these approaches within your instruments. And truth be told, I think the early scanning electron microscopes were mainly used for, I think, in the physical science in order to, for example, observe metal surfaces and stuff like that. So they were actually quite strong in power and so on and so forth. So meaning the use of living tissue, or to use scanning electron microscopes or electron microscopes in general with live tissue is still something which, how can I say, is still even nowadays quite demanding, because you have a quite powerful system, but you also have a very sensitive sample. So and there you also again need to have lots of checks and balances in place in order to make sure that you cannot treat the sample very harsh. So that's why you also have to do the process of the scanning and maybe even of the milling quite slow. But at the same time, it's also so that these systems, I think, initially weren't necessarily designed to operate over a long amount of time in a very stable, with a very stable performance. And I think that maybe separates right now also the systems which we have at Janelia here, because they have been optimized for stability. So you cannot use any, I would say nowadays, kind of any FIB system in the world in order to just randomly scan an entire fly brain, but you need to have lots of feedback loops and so on and so forth in place in order to, and it's again a very lengthy answer. Why is it so lengthy? Because it's quite complicated, and I would say our scientists here, in particular the team around Harold Hess, they've optimized this. They've extremely optimized this so well that I think right now they have a very good handle on their systems. Every time when there's a new sample coming in, they can with a very high confidence scan this and get good results out. But anyway,
Thanks for that. Just complicated, but can be done. Yeah, I think in general.
Yeah, but I do think, like, I think the science which is done here on campus, specifically with these correlative approaches, is, I think, I mean, it's pushed so much to the limits here that I do think that people right now all over the world are kind of right now very eager to implement these tools also in their own labs. And I hope, fingers crossed, that also other companies around the world might actually soon jump on the bandwagon and also reproduce the systems which we have here, such that every scientist in every lab in the world has the capability to do exactly correlative microscopy in such a way as how we as we do it here on campus.
Cool, great. I guess there's one last question from Tong from Olympus. She asked that we have covered very in detail on super res techniques. And to some extent, from her perspective, confocal is still a pretty strong workhorse in most facilities. Which would you expect to permeate it in terms of super res in localization microscope into more apps and facility from your perspective, in terms of like a, you know, probably a microscope in a box?
So the question is like, I have to like, so the question is specifically, what's the go-to system in the future?
I guess so, yeah, for a localization type microscope, what would it be? Because there's a few covered, there's two, I guess Tong is asking about 2 and 3D. You've covered both, and there's a software aspect to that. It doesn't seem to be a straightforward commercial system.
Yeah, I think I'm just like Tom, do you maybe want to unmute yourself and
That's
ask
right,
the question on your
Is she on? Tom, did I cover that right?
Are you still there?
She's around, Tom? Yep. She's not.
Yeah, I'm
Probably
just like but we can like I can still maybe just like which do you think will be the commercial
type ready to go system
in the future? So
sorry. Okay, sorry, it's a bit computer issue here. Thanks, Steve, and thanks. You're right. And then actually, my question is, you give a really excellent report on the single molecular localization microscopy, and then which required special dyes, and then to pick the suitable buffer and etc. A lot of post processing. So my question is that because you are really an expert in this field, and if you look at in the future, what kind of super resolution technology will be developing very quickly and like easy to go, one click, not really one click, but more user friendly for the users who's using the microscopy in the biology fields. So what do you think?
So I think maybe one thing is, I think the underlying restrictions for all these technologies, since this is like fluorescent microscopy, are really the flow force. I think lots of these technologies are kind of pushed to the extremes, but could maybe even perform better when the photophysical properties would be adjusted also accordingly. So, for example, with even in the case of STEP microscopy or resolved microscopy, there it's, for example, so that you can actually also with these scanning approaches reach quite good resolutions when you actually have a switching process which has a longer lifetime. And but there are also not so many fluorophores right now available which exactly how can I say fulfill that niche, and therefore I think which kind of tool, yeah, I mean that's in general like a very big microscopy question, because microscopy unfortunately or fortunately, however you want to see it, is not a discipline which is like driven by just one very specific scientific field, but microscopy is a very interdisciplinary approach, as you've maybe seen, I guess, during the talk here as well. So you have the technological part, you have the sample prep part, but you also have the analysis part, and so on and so forth. So first of all, I do think that the analysis side will, over the next couple of years, actually improve quite strongly. Also, what you've like the last aspect, for example, of the talk with the decode software, where we are actually more and more able to handle big data sets in a more reliable and in a more satisfying way in order to analyze. In order to get what we need, meaning that the big drawback and bottleneck which we had right now in single molecule localization microscopy, meaning the processing, that will be way easier to handle. So that's one part. But I guess also again with the dye developments, you might not need any more crazy dyes and crazy buffers which are entirely not suitable for live cell imaging. So that will hopefully also be solved over the years. And but I mean, I think one aspect is maybe oh god, very cheesy response. I know that I'm having a long responses here, but but I think the technology which is in the best possible way able to capture with the highest integrity biological processes and structures. That's which is I think very broad response, but that really depends on quite a lot. And meaning people always maybe complain about the intensities with STAT, but STAT can kind of follow dynamic processes in a quite good way. But it's also not, how can I say, but there are also other processes, for example, resolved or other kind of mechanisms in particular when you parallelize this technique or when you are way more efficient to get rid of background signals. So it's really hard to predict, because I know that there's lots of work going on in different types of fields. But I would not narrow this down to a specific technology, because they are all also have their specific hardware maybe advantages and disadvantages. So yeah, Steve, you wanted to say something?
No, I was just saying that, just to summarize, I think just extremely complicated, and I think there's chemistry, physics, computing involved, and in general, I think the question drives the process, and if there's a right question, there's a right big question, systems can solve, you know, I think whatever wins to a champion. Yeah.
Yeah, and I think to quite an extent, it also really depends on how these different fields communicate with each other.
Correct.
I mean, what I like here at Janelia about this campus is actually that we do have different people interacting with each other. I mean, Luke's lab, like a chemist, then we have Srini, who's like an expert in machine learning, and again, Harold has with his like very technological or hardware focused science, but these all kind of play together. And if you only work on a very narrow aspect, you will not necessarily be able to push the entire thing for a benefit for everyone.
Fantastic. So I think we've got a lot to absorb and a lot to think over and a lot to and all of this will be on YouTube. So questions, please send Ulrike an email. And thank you all for staying along for this extended time. So have a great evening and a great day ahead for everyone.
Okay,
Thank
bye-bye.
you, Ulrike. Thank you.
Bye.